and welcome to today's In Conversation with Cleanroom Technology. I'm Sophie Bullymore, the editor of Cleanroom Technology, and today I am joined by Miriam Guest, the Associate Principal Microbiologist at AstraZeneca. Nice to meet you, Miriam.
Thank you.
If we could just start with a little bit about the background of microbial identification and why you perform it.
So we're doing quite a range of microbial identification and why we do it for slightly different things, but we'll start, say, with the product-related contaminants. So in the case of non-steriles, we're assessing any counts for the risk of objectionable organisms in those total counts. We might want to understand our in-process samples and our raw materials to understand what those organisms might have an impact on our product or our production areas. In the case of a sterility test failure, obviously that's a major deviation, and we need to understand as much as we can from the root cause. So the microbial identification there is pretty critical. The majority of the IDs that we're performing are from our environmental isolates. So our environmental monitoring program, it takes snapshots of our cleanroom environments. We want to understand what typical looks like, so then we can understand if we've drifted away from that norm. And to do that, that microbial ID is really important. We've then got our personnel that are going into our clean rooms, and we all know that's the biggest risk of microbial contamination in the pharmaceutical clean room. So when we're taking samples from that and looking at the IDs that we recover from there, we're looking to ensure that our training programs and our gowning procedures are effective. Again, much like the environmental monitoring, we're looking at a drift from the norm and trying to understand if we're still in control. But we also want to understand if we've actively compromised any of our products or our production areas and brought anything in that we need better control of. In addition to that, we've got our utility isolates. So are our services being maintained appropriately and correctly? Is it well controlled? So things coming from our water systems, for example, and the sum of all these IDs helps us to understand that. But ultimately, we're working with our regulators to make safe and efficacious medicines. It's really a partnership to protect the patients. And there's a number of regulations that call for us to do microbial ID. You've got the ICH guideline Q9 on quality risk management. We've got the EU GMP Annex 1, and certainly in the draft, the emphasis on microbial ID has been enhanced. FDA guidance for industry, this isn't a new regulation, this is back from 2004. It talks about including the routine identification of microorganisms during that environmental monitoring program. And USP 1116 on controlling aseptic process environments. Again, that talks to the importance of a good identification program. But fundamentally, we're performing these IDs to understand our systems and our processes, our products. Our regulators expect us to, but the bottom line is we're doing this to protect the patient, and that's our number one priority here.
So to go into a bit more detail, could you explain how you use that data and sort of what sort of data you use for an effective program?
Yeah, so the trend in your environmental monitoring program is really important. We want to be able to understand and assess those drifts. So robust trending, understanding where you're recovering organisms, how many you're recovering and what they are is really important to maintain that level of control and respond to events. We're also, when you think of a deviation event where you've got an out of specification in a final product test, you want to understand why that happened and address it. So looking for that root cause. So that final ID from your product on its own wouldn't be enough to tell you that. You need that data that sits around it, your environmental monitoring, your utility and your personnel monitoring. That will all help lead you to a root cause. So the more robust data you've got, you can streamline that investigation and hopefully identify your root cause quicker and resolve the issue. We're also using that data to transform our micro labs within AstraZeneca. Our regulators are expecting us to progress with new techniques to make our processes more robust, reduce subjectivity, reduce lead time to results. So we want to progress with these alternative micro methods. And the major pharmacopoeias give us an outline of those expectations. Both the United States and the European pharmacopoeia talk about demonstrating the alternative micro methods' ability to detect a range of microorganisms. And we have to justify how we select those organisms. So we did a lot of work looking at trends in cleanroom flora to understand what the most frequently occurring organisms were. And this gave us a really robust justification, because we could say what we were most likely to see as contaminants in our product, and build those into equivalency studies to demonstrate those alternative micro methods effectively.
Brilliant. So what do you think are the benefits and challenges of a multi-site approach to ID within a larger manufacturing network then?
So I think in terms of the benefits, that harmonized approach, you get a standardized approach to the level of identification. So, we can talk about ID in different sort of categories. You may just be characterizing your organisms. You may be identifying based on gram reaction, so gram positives or gram negatives. You may be going for a genus ID or a full species level ID. But having a standard approach to what you identify in which situation, that really helps when you're working globally. Having that standard data capture as well, when I'm looking at annual environmental monitoring reports from different sites, having standard graphs or tables, that really does help understand and interpret the data. We also see that we're moving materials between sites, so having a concise data trail of what's associated with that material is also really helpful. And as I said, that implementation of new technologies, being able to have a global picture, does really make for a robust approach to implementing new technologies. In terms of challenges, we've got some regional differences of what expectations are by different regulatory agencies, which is one challenge. And obviously history, we've got very familiar ways of doing things in our labs, and change is always hard. So there may be historical reasons, perhaps an audit observation for why things are done a certain way. So that can also be a challenge.
Brilliant. And so how then is microbial ID linked to the application of data for these new quality control technologies then?
So with the review that we did to support our onboarding of new technologies, we looked at what the most frequently occurring organisms were for different regions geographically. So we were looking at the end game of getting alternative micro methods in a range of labs around the world. So we wanted to make that process really robust so that we wouldn't have any surprises. We know how far we can push it or what range we can get. So having that, we knew that the most common organisms were gram-positive cocci that were coming from our personnel moving into the clean rooms. We also knew what some of those less frequently occurring organs were, but that were also quite high up in the hit list. So a gram-positive rod, we included that in the panel. So being able to do that, we were able to select this organism panel based on the broadest risks of what might be found in the production environment. We had a sound justification for it. So not only did we use independent data from a provider that was doing over 400,000 IDs a year, we verified that back to our internal AZ environmental monitoring reports for the different sites, and it all lined up perfectly. So we had a really robust justification for that organism selection. When we're working to move methods to different labs, we can talk to how we've captured the regional nuances of their flora as well. So you could take it to the next level when you look at seasonal variation, such as filamentous fungi, which we often capture in discrete seasons, and that may help us understand the risk of those types of organisms to our products as well.
Oh, fascinating. And I guess finally, looking forward, what do you think the future of a microbiology lab is?
Well, I think, we've been talking about modernising micro for quite a long time. Some of our methods are still back in the Louis Pasteur era. He wouldn't be that unfamiliar with some of the things we're doing in the micro labs. And you'll see pictures of Fleming in the 1940s holding agar plates that look exactly the same as the ones you'll see in our labs today. But we are seeing new techniques being used. We are seeing more at-line real-time monitoring. We're seeing faster techniques with perhaps molecular or bioluminescence techniques for that rapid detection of microorganisms. And I think as we build these pieces together, we'll be much more streamlined, but also have much more understanding of the control of our environments.
Brilliant. That's all fascinating. I learned a lot. Thank you, Miriam, for your time. Viewers, if you would like to see more interviews with companies like AstraZeneca, head to our YouTube channel or to cleanroomtechnology.com. Thank you for watching.